Well, hello. Good morning, everyone. Thanks a lot for this introduction. And so, well, today I will be actually presenting the result of one of our basically most important collaborations between the University of Eastern Finland, the AI Virtanen Institute, as well as the McGill Center for the Integrative Neuroscience. Basically, as we have seen so far, very important rich multimodal as well as multi-scale data sets as well as atlases have been actually carefully curated and proposed in the case of the adult as well as the fetal, basically human brain, as well as actually the different types of the animal species, right? So for example, in the case of the rats, mice, as well as macaques. And so some of these data sets basically they include only one subject or basically one animal, but then at the same time, they actually will provide a very nice kind of integration basically between different types of the MRI imaging as well as the microscopy modality and techniques. At the same time, for example, some of the other atlases, they provide basically very detailed kind of the average probability maps basically in the case of a population of these species. When it comes to the case of the rats, the story is a little bit different. So, basically in our center, we have been carrying out different types of experiment, looking into the healthy mechanisms as well as different types of the disease models, such as, for example, the traumatic brain injury, the post-traumatic epilepsy, as well as the stroke models. However, in the case of these rats, basically so far, one of the most important kind of the integration has been carried out in the case of the MRI as well as the diffusion tensor imaging atlases. Still, basically, the other kind of the microscopy modalities as well as the histology are somehow missing from this problem. And the question becomes that, well, why does it matter? So basically, as we know, this kind of the preclinical animal models, they basically have the potential to improve the option of the translation, basically, by utilizing very carefully curated data sets. Not only that, basically, all of the new tools and methodology that are being developed basically for these purposes, they can also uncover novel aspects of the healthy as well as the pathological brain. And then, of course, we shouldn't forget the very important aspect of the very careful communication between our own basic researchers as well as the clinicians who can first of all basically guide us, with asking basically the right research questions, forming the right hypothesis, but then also basically explaining the value of these biomarkers in order to improve basically their usability for the real clinical settings. So then, with this introduction, in this study, we had two main goals. The first one was to first of all, provide this kind of the integrative framework in order to bring basically together multi-scale, multi-contrast, as well as the multimodal methods for the analysis of the anatomical as well as the cellular biomarkers in the rat models of traumatic brain injury, or TBI for short. So then, for example, here we are showing the case of the in vivo MRI, the ex vivo MRI, as well as the bright field microscopy. And so the second goal was to basically use the very brilliant latest developments in the case of the BigBrain2 pipeline for basically providing this kind of the systematic co-registration as well as the 3D volumetric reconstruction that would use, first of all, basically the high-resolution multiple contrast microscopy, which has been acquired or basically cut along basically horizontal as well as the coronal planes of these animals. And so basically take them to the space of the anatomical as well as the diffusion MRI at various resolutions. Basically, in this study, our animals were divided into three main groups: naive, sham-operated, as well as the third group that basically underwent either the mild or the moderate, basically, model of the traumatic brain injury, using this kind of a model which is basically called the lateral fluid percussion, which provides basically a very precise impact towards the left hemisphere of the brain of these animals. And so basically here, we can probably appreciate basically the site of the impact that shows, first of all, basically tissue loss, tissue degeneration, as well as the enlarged ventricles. And so actually then, well, here basically our animals underwent basically actually well scans in the case of the in vivo basically 3 days as well as 28 days after the date of the surgery inside our 7 Tesla magnet. And so actually during these scans, we basically acquired the 3D as well as the 2D basically T1 weighted imaging, T2 weighted mapping, as well as the single diffusion encoding. And so for example, here, what I have been showing is basically the case of the the T1 weighted imaging, which actually had the resolution of 250 times the 500 micrometer. And so actually basically on the day 28, after actually this operation, the brain was perfused, fixated. And so basically 1 day later, the two hemispheres were separated. These hemispheres were actually separately scanned inside our 11.7 Tesla magnet, hence the name of the ultra high field basically MRI. And so actually then, at this point, we basically obtained the 3D T1 weighted as well as the single diffusion encoding MRI. And so then, basically here, we are also actually demonstrating basically one of the sections that would actually correspond basically with the side of the impact, again, actually having been obtained from the left hemisphere of these brains. In the case of the mild TBI animals, the voxel resolution here was basically equal to 7 micrometer isotropic. In the case of the moderate TBI, it was equal to 100 micron isotropic. And then, finally, the brains were basically sectioned, mounted, stained. And so actually, basically here, we are actually showing two of the series, basically the Nissl as well as the myelin stained sections that were basically scanned at the basically at the native resolution of 136.9 nanometer per pixel. And of course, for this kind of a data set that basically includes different kind of time points, as well as, of course, multi-scale imaging in vivo, ex vivo, as well as the different kind of the modalities, performing basically harmonized effort was actually very, very important. And so then, because of that, we obviously had to start with the case of the visual quality inspection. And actually, here basically we kind of joke that we had to basically go back to the source data in order to actually identify, for example, what were the missing histological sections, broken sections, as well as, of course, the challenge that would come in the case of the intensity variations. And then, of course, the normal kind of the pre-processing, basically metadata extraction, conversion of all of these the modalities was, of course, had to be carried out. And then, at this point, we basically made the decision to basically downsample our histology photomicrographs to the resolution of 1.095, basically 8 times downsample, in order to at least basically be able to actually save and then, of course, process the data. And so then, at this point, basically, the fabulous semi-automated BigBrain tube-based pipeline actually comes into the place, which was actually so well used in order to basically provide the histology to the MRI co-registration as well as the 3D reconstruction. The current results that we are showing at this point were basically reconstructed at the resolution of 10.95 for the sake of the computational basically feasibility, but then, of course, in the future, we would actually basically be able to kind of replicate the same pipeline into the submicron levels. And so then, basically, this pipeline actually included 5 different steps, starting from the case of the automated alignment or basically centering of the consecutively stained sections. So basically, Nissl to Nissl, myelin to myelin. And then, after that, basically, we carry out the affine registration of our histological block to the ex vivo structural as well as the diffusion weighted MRI maps, followed by basically performing iterative 2D as well as the 3D, basically linear as well as the nonlinear transformations between the stacked histological block and our reference, MRI volume to basically allow us to account for the translation, rotation, scaling, and of course, shearing. And so then towards the end, we also performed the section-to-section nonlinear alignment. And then, at the end, we had to actually carry out the optical balancing of the entire 3D volume in order to basically improve the case of the intensity variations in them. So the first result that I'm showing here was actually acquired from basically one of the animals that underwent the mild TBI, So actually, well, in this case, the left hemisphere was, of course, scanned. And so actually, well, this scan basically actually included everything, starting from the olfactory bulb all the way to the cerebellum of this animal. The cuts were actually along the coronal plane. And then, well, at the same time, basically, the 220 nisal sections that we actually well, had acquired, they basically had a gap of 30 micron between them. So then, well, here, basically, on the left, I'm actually then like, well, sorry, let me go back. Yeah, so like actually like here on the left, I'm actually like showing the results of the linear actually transformations that actually so like well basically like will show actually then like well the very nice match basically between the actually consecutive MRI planes as well as the histology planes, basically actually like we're moving up and down or basically like the front to the back along the volume. On the right, we're actually showing actually so like this kind of the animation of the reconstructed histological block at the resolution of 10.95 times 74 micrometer. And so actually, like with this video that we are actually so like we're quite like we're proud of, basically actually so like it shows this kind of the volumetric reconstruction of this block. Basically actually so like we're showing, of course, all of the actually like the different kind of the imperfections. And of course, actually, so like here, basically in this video on the left, you can actually like basically see the reference MRI block. So actually, like on the right, basically the reconstructed histological block, both of them actually in the same space. That also actually like shows basically some of the challenges in the case of, well, for example, the cuts along the actually so like the cerebellum as well as the olfactory bulbs. And so then, well, the second basically like result actually, so like in this presentation, so actually like, well, here basically in the case of our almost like with the 25 actually, well, the case of the moderate TBI animals, the cut were actually along the horizontal planes, but well, at the time, actually so like here basically the number of sections were actually lower because actually so like here the histology was actually carried out in the manner of the 1 in 5 sections. So well, basically between actually like well, each of the two nisal actually sections, there was actually a gap of 120 micron. That was obviously actually sort of challenging, basically the accurate kind of the basically like the section to section registration. Yes, very quickly. And so actually then here basically in the case of the nisal to the T1 weighted, as well as actually then like for the case of the nisal basically to our diffusion actually well encoding. And then, well, basically, actually, sort of towards the end, the entire kind of the reconstructed actually 3D block that I hope it's visible there, well, maybe not that much. And yes, so then, well, basically, actually, in summary, we have actually so like shown that the BigBrain2 pipeline actually so like well, basically like enables actually so like the integration of the microscale histology as well as the ultra high field MRI at the actually so like 11.7 Tesla, and of course actually so like the results have been reproducible in the case of the coronal as well as the horizontal cuts, and of course in the future we would basically actually hope to be able to actually then like well, move actually like towards actually so with this kind of the resected tumors or the epileptogenic tissues that have been acquired from our own basically human patients. And of course, the current actually sort of the quantitative like well evaluation is being actually well carried out. And then well, of course, actually so well in the future we would like to basically like well go actually then like well down towards the submicron levels. And then, well, of course, that's actually, I mean, well, so actually, like, well, not the whole story. We are also actually, like, well, basically very, very carefully curating our data set. And so actually, like, well, at this step, basically, the MRI, like, the data sets are actually fully compatible with the BEATS actually organization. However, actually, then, well, in the case of the microscopy, we have been actually somehow actually, like, modifying, the kind of organization so that it actually sort of well fits basically our current data a bit better. And of course, the entire kind of the workflow is actually well being basically sort of unified to actually allow the scalability of this project. With that, I would like to sincerely thank all of my collaborators at the AI Virtanen Institute, University of Eastern Finland, and I will, of course, a sincere thanks to PJ, Alan, Claude, and Reza, basically, for actually sort of hosting me for the past 2-3 years during actually my research visits, and of course, the entire BigBrain and HIBALL community. It's been an honor. Thank you, and I would be very happy to answer questions.